Welcome to day 5 of this workshop, which has been fantastic so far. Thanks to all the speakers who have done an amazing job of, telling us about the overview of the subjects and then, of course, the lovely science that they have been doing. The audience has been highly participating, and I hope this trend continues today as well. So to start the day, we have with us Pankaj Alone. He's a professor at NICER Bhubaneswar. NICER is National Institute of Scientific Education and Research. This is similar to the ICERs in India, but by a different heading unit. Pankaj did his PhD from National Institute of Immunology, which is in New Delhi. And after that, he went on to do his postdoc with Tom Dever at NIH. He then had a short stint at UC Davis before he returned to NICER, and he has been there since. He uses yeast as a main model system, and his interests lie in understanding non-canonical translation initiation mechanisms, which I think is what perhaps he'll be focusing on today. So without further ado, Pankaj, all yours.
Yeah. Thank you, Raj. Good morning, everyone. This what we work in translation initiation and to understand how the non-AUG start codons are selected. And we use yeast molecular genetics to understand this kind of phenomena. So as you know that the central dogma of molecular biology culminates with synthesis of a protein. And in order to synthesize protein, you need to establish an open reading frame to decode the genetic information. In the prokaryotic system, IIF2 is the molecule which binds to initiated tRNA. And what you also notice that the prokaryotic mRNAs are actually recruited to the 30S ribosome by the Schreiner-Garneau sequence, and IIF2 then comes and binds to the 30S ribosome, brings initiated tRNA, and then locates the AUG start codon, and then it goes into the elongation stage, where the elongation factor TU brings elongated tRNA, and then it decodes in the elongation process. However, in the eukaryotic system, the AUG codon's location is not fixed. And in order to locate the AUG start codon, it needs a scanning mechanism. And this is actually helped by the initiation factor eIF2, which is a heterotrimeric protein which binds to the initiated tRNA, and then it is recruited to the 40S ribosome, and then it scans and locates the AUG start codon. The elongation process is pretty much similar to that of the prokaryotic system. In this case, eukaryotic elongation factor 1A, which is ortholog of elongation factor TU, binds to different elongated tRNA and do the decoding process. So, if you compare between the prokaryotic initiation and the eukaryotic initiation, what you see that a single initiation factor IIF2 is enough to bind to initiated tRNA and locate the AUG start codon. However, in the eukaryotic system, it has evolved in which there is a GTPase core protein eIF2 gamma, which then binds to eIF2 alpha subunit and eIF2 beta subunit to do this function. If you see elongation factor TU, it has these 3 different domains, G domain, domain 2, and domain 3. They are like packed together to form this tRNA binding site. And if you notice here that elongation factor TU, when it binds to the elongated tRNAs, these are the different contact sites which are shown in cyan color. However, in case of initiation factor eIF2, you see that the tRNA has taken 160-degree rotation, and in this rotation, then it is interacting with the eIF2 gamma subunit. And when it is interacting with the eIF2 gamma subunit, These are the contact points shown in cyan color, which is interacting with the eIF2 gamma, G domain and domain 2, while the contacts shown in blue color and red color shows contacts coming from the alpha subunit and the beta subunit. So it seems that the tRNA is contacting less than 32%. There's only 32% of contact with the eIF2 gamma subunit, and rest of the contacts are contributed by alpha and beta subunit. So it seems that this interaction is probably not stable, and the alpha and beta subunit is sandwiching the tRNA in between it and stabilizing it. So we are interested to understand the intricacies of ternary complex formation. And we are specifically interested to understand what is the role of this eIF2 beta zinc binding domain, which is shown here. And there is a in eIF2 gamma subunit, there is a zinc binding knuckle. I mean, it is like knuckle-like structure is seen in eIF2 gamma subunit, which is actually not seen in elongation factor TU. So if you look close to eIF2 beta zinc binding domain, you see that it is made up of 3 distinct loops. For simplicity, we are calling it as a S loop, which is shown over here. These are the residues for S loop. And this is the R loop, which contains this conserved arginine residue. And then there is a T loop, because there is a threonine residue here. And the structure suggests that the beta subunit may be interacting with the gamma subunit through this guanine nucleotide binding interface, in which these loops are kind of interacting with this GTP molecule. So a mutation in eIF2 beta at the S loop region, which is called S264Y, was already been isolated by Tom Donahue, and he has shown that it has a ternary complex formation defect. And this mutation shows a severe slow growth phenotype. So to understand how this zinc binding domain of beta function in formation of ternary complex within, so based upon this structure, we could predict that most probably the zinc binding domain is kind of involved in beta binding to gamma subunit, and this particular mutation may be kind of a disrupting this kind of a binding. So to test that, we did a pull down experiment, and here you can see that S264Y mutation in beta has 30% beta binding defect. So it seems that because of this mutation, the S loop cannot properly interact with the GTP interface, and it is kind of a highly mobile. And this was also observed in a cryo-EM structure solved by Venki Ramakrishnan. When they took this mutation, they don't see any density for this region in their cryo-EM structure. So then, to understand what is the role of, how the zinc binding domain is functioning in ternary complex formation, we then look for the suppressor mutation. And our strategy is like, we have this mutation in eIF2 beta S264Y clone in luteoplasmid, and then we pass it through XL1 red hypermutagenic E. coli cells. And then we get second site suppressor mutations. And these mutants, then we transform into an E cell, which is already deleted of this SUI3 gene, which is coding for eIF2 beta subunit. And then we look for the colonies which are growing faster than the S264 mutant. So here you see that the S264 mutation shows a severe slow growth phenotype, and we isolated a suppressor mutation T238A. This mutation, so original S264 mutation is seen in this S loop, while the suppressor mutation we isolated is in T loop, where T238 is converted into alanine. So we use ISGenetic to understand translation initiation process. And here is one of the assay we use quite extensively. And this assay was first established by Tom Donahue, where he mutated his four in his for mRNA, he mutated the AUG start codon to AUU. And in this condition, the yeast cannot utilize or cannot translate the his4 protein, and the cells cannot grow in absence of histidine amino acid. Then he looked for the suppressor mutations, and he observed that certain mutations could able to grow even in absence of histidine in the media. And one of the suppressor mutation he observed was this SUI3-2, which is eIF2 beta S264 mutation. And it works in a way that these mutations now started utilizing the UUG as a start codon, which is in frame, and it is also a third codon from this AUG start codon. And these mutants can able to grow in the media devoid of histidine. And so this screen is called as suppressor of initiation mutation, and we also call it as a SUI phenotype. So SUI phenotype is a gain of function mutation in which it can utilize UUG as an initiation codon as well as AUG as an initiation codon. So then we check whether our suppressor mutation can suppress this SUI phenotype or not. And you see that the S264 mutation can grow in absence of histidine, but the S264 mutation partially rescues this SUI phenotype, which is also called as suppressor of SUI mutant. And we can also test this by having his4 AUG LAGZ reporter or his4 UUG LAGZ reporter. So if these constructs are transformed in yeast cells, wild type or a mutant, and then if you look for what is the amount of beta-galactosidase is synthesized using AUG as a start codon or a UUG as a start codon, then UUG by AUG ratio will tell you about the extent of SUI phenotype. And here you see that the S264 mutation has a severe SUI phenotype, but the suppressor mutation is partially rescuing the SUI phenotype. And the suppressor mutation of its own does not show such kind of a defect. Another assay we use to understand translation initiation is GCD phenotype. And this is related to general amino acid control pathway. And general amino acid control pathway is related to GCN4 expression. And the GCN4 expression is controlled at the translation level by these 4 upstream short open reading frames. In a normal condition, when the ternary complex levels are high, the ribosome translate the upstream open reading frame 1, and then after translation termination, the ribosome, 40S ribosome stays bound to the GCN4 mRNA, scan downstream, and then it can initiate at ORF2, 3, or 4. And Leosh has shown in his work that yes, there are some scanning promoting elements present at the upstream region which promotes the scanning. So in this case, what happens that the upstream ORF3 and upstream ORF4 are inhibitory in nature. That means if they are translated, the ribosome dissociates and it seldomly reaches the main GCN4 ORF. So in normal condition, GCN4 expression is always down. However, in the starvation condition, the uncharged tRNA concentration goes up, which causes activation of GCN2, which is kind of a phosphorylated, and this in turn phosphorylate eIF2 alpha. The phosphorylated eIF2 alpha subunit of eIF2 complex, then it binds to guanine nucleotide exchange factor and it's become a non-competitive inhibitor, and it prevents GDP to GTP exchange. Ternary complex is not formed and the ternary complex levels are low. Under such condition, the ribosome scans ORF1, translate ORF1, the 60S subunit dissociate, 40S subunit stays bound, it keeps scanning. Because the ternary complex levels are low, it doesn't acquire ternary complex. and it can only acquire ternary complex once it scan pass the inhibitory open reading frame 4. And then it acquires the ternary complex late, in the late stage, where it can now translate GCN4 main open reading frame. So only in starvation condition you will see that the GCN4 expression goes up. However, it is also seen that mutations which causes lowering the ternary complex level also de-repress the GCN4 expression. And that is what we call it as a GCD phenotype, general control de-repress phenotype. So in GCD phenotype, the GCN4 levels are always high. So the eIF2 beta S264 mutation, which has defect in beta binding, and it also has a tRNA binding defect. So it has a defective ternary complex. And you see that consistent to its defect in ternary complex, it has a higher level of GCN4 express. So it shows GCD phenotype. And the suppressor mutation can partially suppress the GCD phenotype over here. And the suppressor mutation of its own does not have GCD phenotype. We also tested whether there are defects in ternary complex by doing co-immunoprecipitation assay, where we can test the presence of tRNA by northern blot or the other factors by using different antibodies. And you can see here the wild type. So we have tagged eIF2 gamma subunit with the HA tag, and using anti-HA antibody, we are pulling down the gamma subunit. And you see that this is the level of gamma subunit. In a wild type, you see there is a beta, alpha, and lot of tRNA. However, the S264 mutation has beta binding defect, which is like 30% binding defect, very less tRNA binding, but the suppressor mutation has restored the beta binding, but it has barely restored the tRNA binding, and this is quantitated over here. Interestingly, what we observe that the T238A suppressor mutation of its own has a very strong tRNA binding affinity, almost like 12-fold. We also tested what is the level of ternary complex on the 43S complex, and this we perform by cross-linking, in vivo cross-linking by formaldehyde, and then isolating the 43 to 48 complex on 7 to 30% sucrose density gradient, and then we look for fraction 7 and fraction 8, which is predicted to have the ternary complex. And you see that the S264 mutation has a very less ternary complex, but the suppressor mutation, although the protein components are considerably improved, but tRNA is moderately improved. Probably it is enough to partially suppress the GCD phenotype. Then we also looked or predicted that the beta subunit is maybe also involved in tRNA binding. So if you see the cryo-EM structure, this is the T loop, this is the S loop, and this is the R loop. And the R loop residues N at 252 position and arginine at 253 position. They are projected into the tRNA, and it is making a base contact at A1 position, and the arginine is making contact with this G67 and G68 position. So it may have this, because of this mutation, it has this tRNA binding defect. So since we have shown that the T238 mutation has a very high tRNA binding affinity, we then reason maybe if you combine this mutation with this mutation, then it should rescue its growth defect. So here you see that the N2D mutation has a severe slow growth phenotype, and when we combine it to the suppressor mutation T238A, it partially rescues the growth phenotype. The R2A mutation is more severe, but when it combines to T238A, it still partially suppress the growth phenotype. And consistently, you also see that this mutation also has a SUI defect, and they can also be partially suppressed by combining with T238A mutation. It also partially rescues the GCD phenotype. However, the R253A mutation was so severe that it was not fully able to rescue the GCD phenotype here. So what we are trying to conclude here that the R loop and the T loop, they coordinate with each other to form a methionine, to have this better tRNA interaction. Now, the hemo syndrome is caused by different mutations at in eIF2 gamma subunit. And one of the mutation which was observed was I222T. It's in humans. Tom Dever has modeled this mutation in East eIF2 gamma subunit, and that residue is V281. So he has done a lot of mutation. One of the mutation he isolated what V2K shows severe slow growth, and it also shows beta binding defect and the tRNA binding defect. So since our T238A mutation could rescue the beta binding defect, we reason that if the V2K mutation also has a beta binding defect, it should be able to rescue the beta binding defect. So then we combine these two mutations together and we don't see any rescue here. And pull down experiments also suggest the same thing, that there is a beta binding defect, but when you combine these two mutations together, it cannot rescue the beta binding. So it seems that this mutation which is present here may be disrupting this eIF2 beta alpha 1 helix binding to gamma subunit. And the zinc binding domain which has this S264 mutation, maybe it's a part of another binding site. It's kind of a predicted that there may be two independent binding site of beta by which beta binds to interacts with gamma subunit. So if that is the case, then when we combine this V281K mutation and S264Y mutation, which is predicted to disrupt both the binding site, it was lethal. It seems that there are two independent beta binding sites. So that's what we are concluding, that the gamma subunit may have two independent beta binding sites. The site 1 where it is interacting with by this alpha 1 helix, and the site 2 where the beta is interacting via this guanine nucleotide binding interface. And the mutation which is disrupting this binding site 2 makes it quite mobile, and because of this mobility, the tRNA cannot bind stably here and it falls off quite often. But now when you combine this S264 mutation and the hemo mutation, both the binding sites are defective and beta cannot bind, and probably it cannot allow tRNA binding. So we also isolated the extragenic suppressor mutation of S264Y in gamma subunit, eIF2 gamma subunit. The strategy is almost same for suppressor screening, and here we observed two independent suppressor mutation in eIF2 gamma subunit. One was at the switch 1 region where this aspargine at 144 is converted into aspartic acid. So this is the S264Y mutation, and this is our suppressor mutation. And notice the N residue in the eukaryotic system, and there is D or E residue in the archaeal system. The other suppressor mutation we isolated is in this zinc binding knuckle, where proline at 158 position is converted into histidine. And this residue falls in the region which is so variable across all the species. So then we characterize these mutations, and we observe that the N2D, the S264Y mutation shows tRNA binding defect, alpha binding defect, but the N144D mutation, which is there in the methionine binding pocket. In fact, you see this residue is sitting in the methionine binding pocket. And you see that this suppressor mutation rescues the beta binding defect and also rescues the initiator tRNA binding. And in fact, the N144D mutation by its own has a really high tRNA binding affinity. So when we check for the GCD phenotype, we see that the S264 mutation shows GCD phenotype which can partially suppressed by this N144D mutation. What is interesting that although it is having a very high tRNA binding affinity, it cannot rescue the SUI phenotype. So that means whatever the tRNA which are bound, they are not in a right conformation, and they may be delivered in premature P-in conformation, and that's why it is causing the SUI phenotype. So in these mutations, it is rescuing the beta binding, but also rescuing the tRNA binding, but the tRNA is kind of in altered state. The second suppressor mutation we are characterizing here is P158H. So the original S264 mutants grow slow, and the P158H mutation recovers its growth phenotype. And it also rescues the SUI phenotype. And then we checked by the pull down experiment. So what you see that the S264 mutation has the beta binding defect and the tRNA binding defect. Interestingly, this mutation, P158 mutation, exacerbate the beta binding defect and partially recover the tRNA binding. So it's kind of a very puzzling that why the mutation is exacerbating beta binding. So to confirm that this is indeed beta binding defect, we overexpress eIF2 beta subunit in wild type as well as P158H. And you see that the P158H mutation has a beta binding defect, and then when we overexpress eIF2 beta subunit, it fully recovers the beta binding. So it suggests that this mutation is working in a way that it's just ditching the eIF2 beta, and somehow it has increased the tRNA binding affinity. So the role of beta was to support the tRNA binding, but here the mutation is kind of allowing the beta to release, and it is the tRNA can bind if it has a higher affinity. So to confirm that this P158 mutation really has a higher tRNA binding affinity, we did a pull down experiment. So here's the wild type level of tRNA bound to the wild type eIF2 gamma, but in P158H mutation there is a twofold high tRNA binding. And this we also confirm by genetics experiment, in which we have this tRNA deletant strain where out of 5 copies of initiator tRNA gene, we deleted 4, 3, and 2. So this mutation has a severe slow growth phenotype. And this can be recovered by if you overexpress initiator tRNA. But this can also be partially rescued by P158H mutation, suggesting that indeed P158H mutation has a higher tRNA binding affinity. So to summarize, we found that nature has given us 3 solutions here about the tRNA binding defect. The first one is it is rescuing it. The S264 mutation is rescued by having another mutation in the T loop, and the beta binding is kind of restored. Another case, it is restoring the higher tRNA binding affinity, and somehow the higher tRNA binding affinity is also rescuing the beta binding to the gamma subunit. And the third one is even more interesting, that the system is kind of even ditching the beta subunit itself and having higher initiator tRNA binding affinity. So this kind of intriguing to us that then what is the role of beta in this case? And our assumption was that, okay, so some point of time when gamma subunit is evolved, it may have this preliminary tRNA binding activity, and it could have been doing this kind of function. So we, in our lab, we had this mutation D4 403A in the domain 2 region, and previously we have shown that this mutation has a severe alpha binding defect. And that you can see that when you have this mutation, it shows severe slow growth phenotype, and when you do a pull down, it does not bind to alpha subunit. So then we thought, okay, P158 mutation has a beta binding defect. Now we combine with the alpha binding defect and see whether the cells can still survive. And yes, the cells survive. So here you see that there is barely beta subunit, there is no alpha subunit, and it has very little tRNA binding, but the cells still survive. And then, yes, indeed, it's the alpha binding defect, and the tRNA binding defect we did by overexpression of alpha and beta, and it rescues the growth defect. So what we are proposing here that some point of time, the gamma subunit has evolved most probably from eIF2, and during this evolution, the tRNA, initiated tRNA may have rotated 160 degree, and then it is binding to the eIF2 gamma subunit in this orientation, and then the zinc binding knuckle is also evolved in order to interact with the eIF2 beta subunit and the tRNA, and then the alpha subunit then came later, and then they become a very stable complex. And then this stable complex is then later on subjected to the regulation by this integrated stress response. So that's the story we wanted to tell you. And this is my lab. We are a translation control research group. And most of this suppressor screening work was done by our undergraduate student, Ankita and Nikita, Vivek. And the PhD students who contributed to this work is Aranyadeep, Binayak, and Tanaya. And we get funding from Department of Biotechnology and Department of Atomic Energy. Thank you.
Thank you very much, Pankaj, for that insightful talk. I think we have a lot of time for questions. So,
Pankaj, can you do away with beta subunit?
We wish that but it doesn't work. So we try to evict beta, it's lethal. And we have some other work suggests that beta most probably is necessary for eIF2 gamma folding. So it is kind of become, intrinsic for its, for gamma function.
That's what I was getting. Maybe there are some other functions.
Yes, yes.
So very interesting. I have 3 questions. Let's see if I still remember them. So I'll start from the end. So have you checked if by any chance eIF5B, which is a homolog of IF2, is not taking over the role of eIF2 when you don't have alpha and gamma binding to, alpha and beta binding to, gamma. So basically supporting the lack of this primary complex and delivering tRNA to the ribosome by deleting, the N-terminus, which is not lethal.
Yeah, is
it possible?
It's nice hypothesis. Yes, we had to test that.
You haven't tested it? Okay.
No.
Okay. So the other question is, if I recall this correctly, I think Ernst Hennig showed some time ago, decades ago, that if you overexpress beta and gamma, you don't need alpha.
Alpha, right?
So this what you show would explain this in principle, right?
Yes.
So because it was very puzzling, how come?
Yes. So yes, if we can bypass that mutation, bypass the Jeff activity, most probably it will survive. So we think that the slow growth is most probably coming because it's not performing GDP to GTP exchange. That is a bottleneck. And then most probably some, contributions coming from alpha subunit to stabilize tRNA. But that can be done over with. And still, the gamma may still have a rudimentary tRNA binding function and can still deliver the tRNA to the start site, T site.
Okay, and I forgot the third one. So I'll try to remember while other people will be asking.
So this is regarding this 2 gamma mutation, the 2, yeah, which one? 281K. So there you were looking for suppressors, but in the beta, but at the second site.
Yeah.
So you didn't get any suppressor on the
No,
first site?
I didn't look for any suppressor for V281K mutation. It was the suppressor of S264Y, which shows restoration of beta binding. We thought like it may also restore the other defect of V1.
Okay, so you were not actively looking for
No,
suppressors?
no, but actually we are looking at it, but we haven't got yet the suppressors. But I don't think this suppressor will have anything to do with the second side binding site.
And regarding the evolution of eIF2, did you do, in silico analysis to figure out if there are actually organisms which have rudimentary alpha or gamma, alpha or beta subunits, or absence of these subunits in any of?
No, we haven't checked those things.
I have a very general question about reinitiation. So after translating the upstream ORF, is there a minimum distance which the ribosome has to pass through so as to scan downstream or reinitiate downstream?
So Leo shown that there are certain regions where it promotes the scanning. So saying that how much distance the ribosome has to scan in order to reinitiate in GCN4.
GCN4 or any other
ORF.
mRNA.
Distance between these ORFs, or was the distance from ORF1 to GCN4?
So is there a minimum distance between, say, upstream ORF?
I'll answer that.
Thanks.
And I remember the third one. So can you, I was specifically intrigued by the mutations that are increasing the affinity, for tRNA. So can you use AlphaFold or, go to the structures of Tanweer and see if these would somehow rigidify the complex so that it would be less flexible, so you could not get from the P out to P in, which means from this non-accommodated state to accommodated state during selection. So if this somehow can change the mobility or flexibility, because that could explain that even though you bind more, not you, but the eIF2 binds the tRNA more strongly, it still cannot, you still see the SOI phenotype and the GCD phenotype.
No, so the higher affinity is only with the single mutation, not with when you're combining with the original defective mutation. Yeah.
Somehow changing flexibility.
So it doesn't have any SOI phenotype, it doesn't have any GCD phenotype. And what is also important that when we look these mutations on the 48S complex by this sucrosidase, the ternary complex levels and the tRNA levels are almost same. Okay, so this may be only a phenomena for isolated ternary complex. And the moment the ternary complex binds to the ribosome, it is an entirely different complex, and which doesn't follow the higher tRNA affinity rule does not follow there, I guess.
Pankaj, this is not directly related to the question, but to your talk, but related to what Aman asked regarding scanning in a normal context and during reinitiation. In reinitiation, we understand that the ternary complex is not there. So how does it scan? How the complex scan when there is no anticodon to actually look for the codons that it is going through?
Yeah, but does it need the ternary complex to look for it? Just scan.
So it's actually just moving along.
It's just moving along. Yeah.
It doesn't. It's just. Yeah.
It just moves on from 5' to 3' direction.
So it's not actually checking the codon.
It's not checking anything, yeah, because the tRNA is not there, so it's not checking anything. Just drifting from 5' to 3' direction.
Any more questions? We do have time. All right. If not, thank you once again, Pankaj. The next speaker is Ramu Vemanna. He's Associate Professor at Regional Center for Biotechnology, Faridabad, which is the northern part of India. He has a Bangalore connection. He did his PhD from Bangalore University and then did his postdoc from Noble Research Institute, Oklahoma. He's one of the few biologists, plant biologists in India who is looking at translation control mechanisms. So I believe he'll be discussing with us translation control mechanisms in context of plant development and drought stress adaptation. So.
I'll try. So, good morning, Raj. Thank you for very much introduction. And yes, I belong to Bangalore, not exact Bangalore. You might have all know that Kolar Goldfields. I come from Kolar Goldfields. But I spent a lot of time in Bangalore for studies, basically. After that, I moved to Oklahoma, Nobel Foundation, and then came back as a Ramanujan fellow. It's a privilege to give a Ramanujan Hall a talk, and I should be very thankful to the organizers for giving me this opportunity. As Raj said that my training is from plant physiology background. Somehow I developed interest in the plant translational biology. So I'll be giving a little bit of glimpses what we are trying to do. So all these days we heard about the protein translation and a lot of proteins involved in the synthesis mechanisms. So the major motto of my translation area to get in was the energy production, right? The ATP. So plants develops ATP through rubisco. Rubisco is the one which takes carbon dioxide and oxygen and converts into that. And then ATP is generated there more actually. And then utilized 80% of that into the ribosome biogenesis or the protein synthesis. So my question was very simple. How much of ATP is utilized in that mechanisms? Because plants also has to withstand the stress mechanisms. It's not only the pathogen, it could be the drought or your salt, anything else. So that's how I started, but I'm not doing that work presently. But what we looked into, some of the extra ribosomal functions of ribosome biogenesis factors or the ribosomal proteins, are the people also call them as moonlighting functions and all. So I'll be showing some of these things, and the crop which I work on, rice. So most of you eat rice every day, right? So because rice is one of the food which can be easily digestible by our gut system compared to the pulses and other things. However, growing rice is very difficult task, and to grow 1 kg of rice, it takes about 2,500 to 5,000 liters of water. And India exports a lot of water unknowingly to other European countries, right? So that is a fun part of it. But anyway, so, but what is more concerning factor was what we are eating, the protein content in rice is only 2%, but medically suggested protein content requirement is about 7 to 8%. So, but if you see this one, the varieties which has been bred all over the globe, the protein content has reduced drastically. That means the breeding has been very successful to produce more food without giving attention to the nutrient quality of the foods. And that has led to till malnutrition is we can see in different countries, even including in some of the parts of these things. Although they are affordable, but malnutrition is seeable because of this kind of traits. And then, you know, because of youths are more interested to develop muscles and other things, the proteins are required. So protein content is required. So protein diet is more and more popular in all those things. So my interest was in the drought stress improvement of the plants under drought conditions where water potential of the cell goes down. When water potential goes down, the protein synthesis get affected, and that is due to there is ROS, oxidative stress, or RCCs, or they affect on the protein function or stability, and also it can degrade the membrane stability as well. To maintain all these things, we required a protein to be continuously synthesized. You can imagine this one. This is the normal cell where the ribosome biogenesis is continuously happening. Per minute, there are about 2,000 ribosomes are synthesized. It's a very highly robust mechanism. It can synthesize, but the regulation of ribosomes have not been studied very well. So, but when there is a water is reduced, cells can shrink very drastically. You can imagine Bangalore traffic, you get choked up, right? The same thing will happen in the ribosomes also. The ribosomes may not be functional. So the shrinkage will lead to that one, and this will also lead to the dormancy of the ribosomes. You can imagine the seed, whatever the seed we are going to eat, you are eating in the home, any kind of seed, not only rice seed. It is all dormant seeds. There is a molecule called dormine. Now it is called as a abscisic acid that has been known for the dormancy. We do not know that whether that dormine has any connection with ribosomes. Now we have some clues towards that. Yes. And then this is what we are trying to understand all those things. So in this context, the rice at flowering stage is highly susceptible for the drought stage. Very simple, small amount of water shortage will lead to the drastic changes in the productivity of the cells. So we wanted to understand translation ability of the rice genotypes, whether they have translation is happening or not. And we selected two contrasting genotypes, sensitive and resistant one. And in these plants, we isolated ribosome profiling. We isolated ribosome profiling from both the rice genotypes. What we see that, yes, there is a ribosome profiling is affected, but in the sensitive genotype, I'm sorry. So the sensitive genotype monosome to polysome ratio got affected, and the resistant genotype could be able to maintain better. And we trapped all these polysome-bound mRNAs, and interestingly, the polysome-bound mRNAs in the resistant genotype has accumulated more than the sensitive genotype. And in the stress conditions, the accumulation of polysome-bound mRNAs are more higher number, and more number of genes got accumulated in the polysomes. In the polysomes. And what we see, the interesting pattern, I'm not showing the complicated image of the p-site analysis, all those things and algorithms, but we see that in the well water condition, ribosomes are distributed on the mRNAs very well. Whereas in the drought condition, the ribosomes are accumulated at certain points. What we do not know whether what could be causing this kind of stalling of ribosomes, whether it's an RNA secondary structure is causing ribosome, or whether those dorminins are affecting, controlling on those things, we do not know now. So we are behind that questions to understand what kind of mechanisms could be contributing those things. And all of you may be knowing this photosynthesis of Z cycle, the photosystem 1 and other things. Many of the genes which are responsible for these things are accumulated in the polysomes in the resistant genotype. And in the sensitive genotype, it's not accumulated. And some of these, the late harvesting complex proteins or pyridoxine reductions, you can see that this is the sensitive genotype IR64. The number of proteins or amount of protein synthesized is very less compared to resistant genotype. So this is where what we are interested, how could be ribosomes could be contributing for it. So as you all know that ribosomes are 32 and the 44 number of ribosomes, we know that one. But in the plants, there is a lot of parallax existed. Like yesterday, Sunil Shetty was giving a very good, I'm really sorry for that. My throat, because of Bangalore weather, I got some kind of allergic cold. I'm not able to come up back. I'll be fine. Yeah, anyway, so this is what the complications in the plants now. So there are about 237 large subunit proteins were being identified, reported supposed to be, and 230 small subunit. At that flowering stage, what we saw was about 195 the control and stress 190, and also the small subunit proteins also. These are all detected in the polysome profiling. And but there are more than 35 or 25 genes were more than double, twofold upregulated or accumulated more. Now we do not know exactly what is their contribution for the translation or the plant growth and development, whether these proteins are contributing for heterogeneity in the ribosomes, or they could be doing a specialized functions in the ribosomes, or now the Grub Muller was telling the other day, the complexity in the cryo-EM structures, the where heterogeneity is possibility. Now the in-cell heterogeneity, how do you determine? This is the biggest challenge for all the structural biologists is going to be to solve those things. Thank you so much, sir. Thank you. Yeah, anyway, so what we saw that in the ribosome, the polysome profile data, the differential proteins got accumulated, And RPL10 is the one which we are interested. I'm going to show some of the data. And the resistant genotype got less accumulated, whereas sensitive genotype is highly accumulated, suggesting that the sensitive genotypes required to be synthesized more proteins. That's why more number of transcripts are accumulated in the polysomes. So in the rice, we have seen that there are about 4 to 7 copies of RPL10s. We see that. And in the Arabidopsis, there are only 3 copies, RPL10A, B, C named. They're highly closely homologous, about 80 to 90% of them. And to our surprise, there are another 2 more proteins there in the mitochondria or the chloroplast. And there are other proteins, whereas in the humans, only 1 protein of RPL10. Saccharomyces is also only 1 protein of ribosomal protein L10. So now we can see our complexity, what could be our life in the plant science, right? So then we looked into their profiles, just mRNA accumulation in the polysome bound RNA. The resistant genotype could be able to accumulate more. And this is what we came at now. Now there are a lot of heterogeneity. It could be from the rRNA variations, or it could be from the paralogs, or the structural stoichiometry, or could be ribosome accumulation factors and all. So in all these way, the different projects are in progress at the lab. I'm not going to talk about all of them. So, but what we proceeded further was to understand the function of RPL10 family genes. So we cloned rice RPL10 family genes, and 10A1 was localized to mitochondria, 10A2 was chloroplastic, and 10B, again, as another complexity, it produces 3 alternate transcripts of the same gene, and it also localizes to the nucleus and also cytoplasm. This could be the one could be forming the ribosomal thing. So we don't have answer yet whether all of them are forming a complex with the ribosome or not, right? So that's where I interacted with Tanweer, whether can you able to help me to solve this problem. But he explained me the complexity is very difficult. So, but anyway, that's one of the motto for me to come here to this conference to understand more on these ideas. So we developed a CRISPR lines and overexpressor plants of these things. Interestingly, each of them are contributing differently for the seed development phenotype, and some of them are very lethal. We could not able to develop anything. And even though some of them we developed, they were not able to form a seed. So that means that there is a specialized ribosomes in the context of plant development as well as plant adaptation to the environment mechanisms. Or we can say whatever the food we are eating, it could be specialized ribosomes, right? So that's what is happening. And 2 students now presently, Satyasilan and Manisha is working on 2 different projects on 10A1 and A2 on Satyasilan because there are organellar. This is the wild type phenotype, and this is the phenotype of the CRISPR plant and also overexpressor plants of the A2 and A1. And Manisha's plants are flowered early in the North Indian part of Punjab region. The stubble burning is very problematic because of the duration takes for the harvesting time to the next winter wheat cultivation. So our idea is to identify the early flowering genotypes. And this plant was flowered almost 60 days. It's supposed to be 150 days. And however, what we see here, I saw it could be a lethal phenotype, but interestingly, the seeds are filled up very good. That means how these ribosomes are specially making the plant life cycle accelerating faster. We don't have clarity, but the seed filling is very good. That means that there must be a nutrient uptake and the protein synthesis could be happening there. Or this ribosomal B4 could be a negative factor for many of the ribosomes or the translation factors to be suppressing those things. Or it could be a source sink relationship from the leaf to the this one. So all those things we are exploring those things. This is what our PhD program, and we are trying to identify that are there a specialized functioning. So we will be doing the ribose sequencing for each of them because now we have a independent mutants and overexpressor plants of them, and we should be able to tell more story about the ribose sequencing data, how the large subunit complex is also involved in selective translation of mRNAs. Because most of the time we talk about the small subunit only contributing for the selection of mRNAs, right? So this is another thing what we are going to do in these things. About the dorminin, what I was talking about, the abscisic acid is the one which has been showed where the CRISPR plants or the guide RNA mutant plants are insensitive to the ABA, abscisic acid. and the overexpressor plants are sensitive to the abscisic acid, both B3 and B4. Same case in A1 and A2 also. I'm not showing those things. Now, we did one in vitro assay. The Darwinin inhibits the in vitro translation assay. So at which stage the ABA is stopping, we do not know. And this shows that the RPL10 could be one of the target of the Darwinin. So we do not know. Now we have to see those things. So we will see the aspect of those things. Then further, our interest further elongated that, okay, what could be the extra ribosomal functions of those things. Then we conducted the screens of RPL10 and we identified several ribosomal proteins. And also there is another interesting protein, zinc finger protein, is involved in these things. We heard about Umesh Pillai, about the M6A modifications. I will also be talking a little bit about that. And another ABC transporter protein where CCR04 complex is a homolog of that. This does not have a transmembrane domain. There is involvement in the ribosome mechanisms. And another protein, rRNA processing factor. Shobna is here. She has a poster too, so you can visit that to know more about that, what could be the function of those things. So coming to C3H zinc finger protein, this is localized to the nucleus as well as cytoplasm. And all the different copies of RPL10s interacts with the C3H zinc finger protein. And 10B4 as 10B1 has a strong interactions with that. What was interesting was the interactions were highly seen in the nucleolus, and you can see that the granule kind of things, each of them has a different protein, different functions. So different levels of transcription or different levels of RNA processing, it could be contributing for that. And we also developed again for those plants, the CRISPR plants and also overexpressor plants, and the mutants are susceptible to the ABA. What was interesting was the C3H zinc finger is an M6A writer protein. And we can see that the M6A levels in the CRISPR plants were downregulated. This is the ELISA assay. The same thing in the dot blot, we could confirm that. And the seed filling rates is also same thing, the same phenotype is correlating. Now, the RPL10 interactions with C3H zinc finger with M6A modifications, is there anything to do with that one? So these C3H zinc finger proteins are known to be both DNA and RNA binding proteins. And we did a whole genome chip sequencing data, and we found that they are involved in the transcription start site, both promoter near and after. And we identified several photosynthesis associated genes and transcription factors are the targets of these things. And then RIPSIC, this is very interesting thing what I am showing. We identified the enrichment is more in the CDS region, but that enrichment is highly enriched in the 5' regions of the transcript in the whole genome. This is a contradictory of M6A modifications, what Ramesh Pillai showed that the what's the M6A modifications supposed to be in the 3' UTR regions, lot of them, right? But what we saw that in the 5' region. So then we are looking for the literature, what could be doing those functions. The SARS-CoV-2, one of the NS9 transcript undergoes cap independent translation mechanism that is contributed by M6A modifications. That triggered me to solve the puzzle that RPL10 could be interacting with C3H zinc finger to initiate the transcription initiation through cap independent mechanism. And to surprise, we found a lot of rRNAs and tRNAs for the modifications of M6A modifications. So since it's a writer complex proteins, yes, this homolog of the same protein in the humans has been shown to show that yesterday what Ruchi talked about, the same residue has been modified by the same homolog protein, zinc finger 56, was involved in modifications of those things. So these are the open questions still we need to answer several of these things. And now we could be able to answer all these things. So to go further, what could be their contribution in the transcription? We took one of the promoter, L1 promoter, we made a reporter construct and all. So what we found was C3H zinc finger is a repressor of transcription. And along with that, 10A1, 10A2, if they're co-expressed, the 10A1 is a repressor again, 10A2 is a co-activator based on the repressor activity. And then 10B1, each of the copies are showing different activities. 10B1 is a repressor, 10B3 is an activator, and 10B4 is a repressor. 10C is a strong activator of the transcription. So now we are making the RNA reporter constructs of understanding M6A modifications, and mass spec data is under process. I will be showing maybe next another meeting of those data. This is the end of this story, what I'm telling you about the RPL10 with C3 zinc finger. So I'll be moving on with the RPL10 interactions with RPF2. So eukaryotic ribosomes, as Sunil showed yesterday, 118S in the small subunit and 33 proteins in the large proteins are required, and 5S, 5.8S, 25S rRNAs, and about 49 proteins are required for these things. There are only 3 ribosome structures in the plant till now available. The first one is also my collaborator, the Rajyaguru from Germany. When I joined RCB, I was discussing with him whether he be able to solve these things. Prem Kaushal could not solve that one, but he solved this structure. 2023 is the first one solved. And the second one is from Dr. Tanweer here. So, and the another one was the tomato one. Only 3 ribosome, complete ribosome figures what we have now, till now, in the plants. So anyway, to cut next further, the ribosome biogenesis, how these rRNAs are made in the plants. So it's very complicated mechanisms. There are about 750 copies are tandemly arranged in the plant in one chromosome. We ask a lot of questions which we don't have answer for that. But again, the promoters are highly variable, but the final figure, the final 18S, 5.8S, 25S are completely intact. There is no variations, but there are the variations in the 3' ETS sequences. In Arabidopsis has been very well documented. And the 5S rRNAs, about 225 to 1,000 copies are known. The 5S RNAs are transcribed with the polymerase 3. And within the internal promoter sites of them, there are a lot of variations what we identified, which I will show you some of next figures. So here, for the students or some of the few people, the ribosome biogenesis, how when the RPL proteins or RPS proteins loads onto the rRNAs. So there are a lot of steps involved in these things, including the endonucleases and exonucleases, and also one of the methyltransferase fibrillary, which involves in the methylation of rRNAs, which is also the first protein which will load it to the transcription factors TF3, basically. And then ribosomal proteins load at different stages. And RPF2 loads to the rRNAs at the later stages. I will show you the next figure. So this is from the yeast. This belongs to IMP4 family. And this 90S transcript is being processed further along with 5S rRNA for the production of 25S rRNA, all those things. And that is where RPL10 also loads with the RPF2 complexes along with that, and RPF2 interacts with the NMD protein. It's not a nonsense mediated decay protein, but another protein, NMD protein, was involved to export to the cytoplasm. This is where the assembly happens, these things. RPL10 is the one which also assembles both large subunit and the small subunit in the stalk region, and also RPL10 is involved in the rotation of ribosomes as well. So if you cut these processing bodies, you will see this kind of different processes, just to show that on 45S, 35S, 33, 32, 27, all these things. These are really complex mechanisms. In plants, there are 4 pathways has been identified. Just last few days before I saw in drought conditions, one of the Chinese group has showed that there is additional cleavage site in the B1 to A2 site that is contributing to produce a different pathway of rRNA biogenesis. Right? This is one complexity, whether these biogenesis, whether really contributing for the biogenesis rate, we do not know yet. And what has been known in the yeast, it's at least the RPF2 interacts with RRS1 and RPL11 and RPL5, and that will become a complex of these things. They undergo different modifications. So structural biologists still need to capture different stages of ribosomes or the subunit of the ribosomes. With the cryo-EM help, it could be possible now. Whereas if you deplete RPF2 or any of these complexes, including 5S rRNA, the complete small subunit is stopped and the cells become lethal. That's what expected. So with that notion, we identified this RPF2 in the plants as this is a Brix-2 domain protein, and there is a small domain region where it has a small sigma 70 factor. Sigma 70 factor is known to initiate the transcription in the prokaryotes. So we do not have a lot of information about the sigma 70 like proteins in the plants, but it localizes to the nucleus and all. And here also we identified the interactions of RPL10 with other RPF2 very well. And the interactions were seen in the nuclear bodies, nucleolus regions, and other things. Yeah. So in rice, the rDNAs are being distributed in 2 chromosomes, chromosome 9 and 11. This is a fish assay to show the one. And whether all these rDNAs are upregulated or activated at the same time or not, we do not have clue. But here, one of the study in the Brachypodium, you can see that this is the parts they have taken throughout the life stages of these things. And if you see this aspect, the pink colored one, these are the 2 genotypes, the expressions of rRNA production. rRNA production is different at different stages. That shows that the specialized ribosomes exist in the plants from the day of their birth till the day of their death. So all stages, there are different ribosomes accumulated. So if you solve a structure of ribosome at one stage, it may not be same thing for the another stage. It could be. That's another complexity is coming up. So this is also true in Arabidopsis. The rRNAs, which is expressed in nuclear organization region at chromosome 2 are inactive, whereas at the developmental stages, the nuclear region 4 will become active. And how they are distributed. This is our data suggesting that there are 4 regions we identified in the chromosome 9 and 11. And you can see here, there are some white patches here. This one figure I'm trying to show that there are a variability in the rRNAs, 5S rRNAs. Although it is involved in catalytic functions in the P site where peptidyl transferase reactions happens, right? So, but how each one is contributing for what kind of mechanisms, what speciality they are bringing up, we do not have any clues about those things. So we developed Arabidopsis overexpressor and mutant plants. The overexpressor plants are very good. We expected the mutant plants supposed to be lethal, but it's not lethal. I don't know the reason because IMP4 family proteins, there are 4 of them in the plants. They could be taking over on those things. There could be redundant functions of those proteins contributing for rRNA functioning, but the modifications or whatever it's contributing, if the mutant plants are, which are very, a little bit lesser in the phenotype, there could be specialized ribosomes could be forming up those things. And we see that phenotype, the stomata phenotype is much more bigger than the RNA plants. We quantified that adaxial and abaxial leaf surfaces, all those things. What as another interesting phenotype was the trichomes. So in the deserts and other things, plants develop an adaptation mechanism. Trichomes are developed to save the water mechanisms or water, or also to protect against the herbivores or pathogens and other things. And we see that their trichomes are early, more developed in the overexpressor plants, whereas in the flowering is also early flowered. So I showed RPL10, there are some mutants are early flowered, here the RPF2 overexpressor plants are early flowered. So they are differently functioning in the regulation of plant development. So we have these 2 plants, overexpressor RPF2 and RPL10 plants, and we did design some probes on the 45S rDNA, but we designed the probes on the mature 5.8S, 25 and other things. You can see here, the RPF2 overexpressor plants and RPL10 overexpressor plants has a good accumulation of rRNAs compared to the mutant plants and other things. That is contributed to the total protein synthesis also. The overexpressor plants could able to synthesize more proteins, whereas RNA or the mutant plants could not synthesize more proteins of these things. So what could be their role? We don't have any ideas about that. And then we subjected for the drought stress, and these plants are very highly tolerant to the drought stress, whereas mutants are not tolerant to the drought stress mechanisms. And we did a whole proteomics analysis. The proteome, interestingly, the wild type plants are not behaving similar to the overexpressor plants. If RPF2 and RPL10 both are involved in protein synthesis alone, there should be an equal same pattern of protein synthesis or same protein accumulation should have been there. That pattern we did not observe in both the RPL2 and this thing. But however, we did a different patterns of those accumulations. And both the overexpressor plants showed different pathway genes accumulated on those things. That means there is a unique set of proteins are translated by the different proteins, RPF2 or RPL10, even though they're interacting. They could be having some functions in biogenesis, all those mechanisms. So that means that they must be having extra ribosomal functions. This extra ribosomal function could be attributed to RPL10 with the respect to C3 zinc finger protein. RPL10 in the humans also known that it is involved in interaction with the C-gene transcription factor, whereas RPL10 in plants also interacts with other MIB transcription factors as well. Whereas the RPF2, being an rRNA processing factor, it did not confirm that whether it has any extra ribosomal function or not. So we identified again the interactors of RPF2. We identified the MADS transcription factors, and these MADS transcription factors operated by polymerase 2, whereas RPF2 supposed to be with the polymerase 1. But now we saw the interactions of those things. We see the interactions in the nuclear, RPF2 in the nucleolus, whereas MADS transcription factors in the nucleus. Whereas they are interacting, MADS 50 interacts at the nuclear ring, whereas the 56 interacts at the nucleolus region. So then what could be their mechanisms? rRNA transcription initiates at the nucleolus region, whereas the processing happens in the nucleolar organellar region. There's the dense fibrillar regions. So whether MADS 50 also involved in the processing of rRNAs, we did not know, but we identified some of the signature cis elements on the rRNA motifs and all. We are trying to figure out what could be their functions on those mechanisms again. So this is the confirmation for interactions to show that biolayer interferometry assay, that this, when we mutate the sigma domain, the interactions came down, but interaction is still there. It did not still vanish. That indicates that sigma domain may not be functional for this. We are in the process of developing more mutations on this area. And then once we know that transcription factor, whether the RPF2 has any DNA binding activity or not. So I'm sorry, okay, another transcription factor. So this is a NAC transcription factor. It's only plant specific. This is not there in yeast. It's not there in humans as well. MADS transcription factor also, it's not there in the mammals or in the yeast. So these are plant specific proteins. So whatever we understand in the yeast, it's not good for me, because in the plant, it's not working the same because it has extra ribosomal functions and it has additional role. So what we see here, same thing as a pattern of MADS transcription factors, NAC transcription factors also, we could see that one of it is the interacting in the nucleolar ring, whereas the another one is interacting in the nucleolar ring as well as in the nucleolus region for the differential regulation. Although the homology between these proteins are very homology, about 80% close homologous. So to explain that phenomena here, the transcription of rRNAs happens in the nucleolar region. It's a dense fibrillar region, whereas the rRNA processing happens at this region, and granular components, what we see here in these regions and all, are involved in ribosome assembly, because RPF2 is known to be involved in ribosome assembly. What is not known was whether these transcription factors are also playing a role in ribosome assembly is not known. Whereas they are only localized to the nucleus, or they are also exported to the ribosomes, through the ribosomes to the cytoplasm is not known for us now. So we identified whole genome transcript, the chip sequencing analysis, we identified signature motifs and all, and blah, 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 all these things. And then we identified several different pathways are regulated by these proteins and all. Many ribosomal proteins are targets of RPF2. And we confirmed their interactions by EMSA, one of the promoter, RFT promoter, that is involved in the flowering mechanisms. So one of the gene which is regulated by RPF2 as well as MADS transcription factors. And we constructed the reporter construct. And what we saw that when each of them have a transcription activity, all these bars shows that they have transcription activity. Whereas when they are co-expressed together, RPF2 and MADS transcription factors, there is a suppression in the transcription. So how the suppression is happening, we still don't have any clarity about that. We are doing some experiments on those areas. So subsequently, the interesting thing was another NAC60 transcription factor. The targets were not known. So we identified the targets. We identified some of the cis elements and all. So what was more interesting was both RPF2 and NAC60, both of them binds to the same region for the computation mechanisms. Whether in the one of the chromosome, what we see here, you can see the peaks, the enrichment of the proteins on the same regions, and we identified several proteins or several genes which are commonly regulated by both the RPF2 as well as NAC transcription factors. So now, whether they are acting as an inhibitor or they are acting as a co-activators, we don't have clarity about them. We are figuring out all those aspects. So we developed NAC60 CRISPR line as well as overexpress line. Shobna is here. She have a poster. You can visit that. She will be explain more. And these plants are also early flowering. The CRISPR knockout plants are early flowering. If you remember, I showed in Arabidopsis that overexpressor plants are early flowering. So that means there must be a competition for the flowering regulatory proteins. And these knockout plants, CRISPR-edited plants are also drought tolerant in all these conditions. This is what I have for the plant development and drought stress adaptation mechanisms. So overall, we could be able to show that at least the RPF2 interacts with RPL10 for the processing of RNA biogenesis mechanisms, and RPL10 could interact with C3 zinc finger proteins for the rRNA processing, and RPF2 may interact with NAC or MADS transcription factors. They're all contributes for the overall translation mechanisms in this whole thing, and that could be contributing for plant growth or ribosome biogenesis or drought tolerance. What we do not know yet is grain protein content. What could be their fate? We don't have it. Maybe in the other meeting, I will show those aspects of this. With this, I would like to thank our group. And most of the work, what I showed, was performed by Akshata. Now she's not in the lab, and now Manisha and Satya is taking over. And Shobna is here. Shobna will show some of the data with RPF2. And thank you very much for listening to the plant biology. Thank you.
Thank you, Ramu, for that wonderful talk. Again, he has finished very much on time, so we do have time to take a few questions.
How many different genes are there? You mentioned a few, I think.
Four.
Four, and they have some heterogeneity in the sequence there. Okay, and RPL10 is, I think, located at the interface between the
large subunit and small subunit.
So have you looked at any aspects of the interaction between the two subunits?
No, that's what I'm asking Tanweer to help me to look at that. What could be their mechanism?
But you do see that there is a sort of pattern at which stage of development one will come and then the other one will take over.
Yeah, but all of them are there in all the stages. But although there are variations in the levels of transcripts, but all of them are there to move.
So if you knock out individually, can you find out which stage is affected?
Yes, now I can say those things, because with the CRISPR knockout plants, we have a different phenotypes. With that phenotype, we can go for the solving those structures.
I see. And RPF2, that is working as a biogenesis factor.
Correct.
Also as a transcription factor.
Transcription factor. Also now what we see that it has a binding site in the internal ITS2 region. So now I am also trying to see whether that ITS2 or anything is contributing along with the polymerase 2 is involved in any of transcription mechanisms of those.
So when you're saying it is working as a transcription factor, it binds to the promoter region, or it is by overexpression only you are telling?
By overexpression only I'm telling. But
how do we know that it is not an indirect effect?
But we see the signatures of those things in our chip data. We see that signature sequences on those places. The RPF2 is enriched in those places.
In terms of binding to the promoter, was my question was.
Yeah, they bind to the promoter. We saw that in EMSA and all.
Hi, sir. Nice talk. I have couple of questions.
Mhm.
So, first is you mentioned that these rRNAs have heterogeneity based on the stages they are in.
Mhm.
So,
Yeah, they see the heterogeneity in different parts as well. Mhm. Okay. Only selective rRNAs could be involved in the making of specialized ribosomes, or I can say unique set of ribosomes. Uh-huh. Yes, we saw the sequences where the A sequences are there. So we also looked at the cap, M7G cap. After that, there is an A is coming up. Also, we are looking for whether ATG is also contributing for this M6A modifications. No, that's the whole genome, the 5' enrichment, what you are asking, correct? No, I did not look for the ORF1, but what we saw that in the ribosome accumulation in our profiles, there is a pattern of some places where we see at least few transcript what we saw that there is a secondary structures what we saw. And those secondary structures are rich with the GC motifs. So we do not know what GC is contributing for that. So there are about 8 nucleotides what we saw there. Yeah.
So I'm wondering, in terms, I guess I'm not sure how you're thinking about specialized ribosomes, and I'm wondering, can you follow each of these different, RPL10 variants with
Yeah.
antibodies? And can you then see what fraction of the protein is incorporated in the ribosomes to distinguish a role in the ribosomes versus in transcription or biogenesis?
So now I have a mutants of each individual protein mutants I have. With that, I can do ribosequencing, and that I can say that which of those transcripts are translated. But how much of proteins are synthesized, that we need to do it. That's a good question.
Right. So I guess but can you follow what I want to know is what's in your ribosomes? How much of the RPL10 is in the ribosomes, each of those species, and how much of it is
we were
free?
not successful. We tried to attach some tags. Our GAP tag is about 30 kilodalton and it screwed up and we could not see in our profile. Even in the ribosome profile itself, I have a fluorescence profiler. I could not get that one in the tagged version. Now I have a low protein, which is about only 10 kilodalton. Now we are tagging with those things. I wanted to show that question, that answer. Which of out of all this, which one could be a part of ribosome?
Yeah. I mean, it just seems that you're proposing many roles for RPL10.
Yes.
And roles in transcription can easily explain.
It's a mystery protein.
You don't need to invoke different ribosomes. It could all be the result of the other roles of this protein.
If you remember the mathematics NCR question in permutation combinations, with all this, there could be lots of ribosomes will come, right? I calculated once, it comes about 30 lakh different ribosomes with all the paralogs of those things. And that could be contributing whether they are having a selective translation or preferential translation could be doing that. But more mysteriously in the human, there's only one RPL10. Right? That is doing a lot of functions in all types of cancers, also involved in cell proliferations, breast cancers, prostate cancers, everything. So I still don't have any good say to say this is the function it's doing. It's doing everything. Yeah.
Variants of RPL10, right? And then it also interacts with different proteins like RPF2 or this CH3 zinc finger, and then they do different function. Do you think that these different variants can be involved in these different interactions?
All variants interacts with all of them.
All variants.
At least one of the protein, two of the proteins with C3H zinc finger and RPF2, what we see, all those variants are interacting with them at different strengths.
Okay.
Yeah.
So the other thing is you're talking about the methylations, right? Is that on the ribosomal RNA or in the mRNA?
mRNAs are more enriched. Ribosomal RNAs, only one residue in the 25S rRNA is known.
Okay.
That has been already, in the human homolog, that is a exosome 56, it's been named as, and that is involved in recruitment of methyl transferase. That is where yesterday Ruchi was presenting. I was contemplating whether methyl transferase alone is doing that function, or some of the reader complexes are required for rRNA modifications and also tRNA modifications too. So I have selective tRNAs. So work is happening in that direction. Maybe we'll see that.
Sounds really cool. From structural modeling, is there any way to tell RPL10 has any kind of DNA binding domain? Any similarity?
RPL10 don't have DNA binding domain, but 1970, one of the papers showed that it has a DNA binding domain. We did EMSA with some of the selective things. We are not successful.
Could it be that we don't understand, we don't know the full complement of what DNA binding domains look like?
After that 1970s paper, I did not see any paper saying RPL10 has any DNA binding domain. But what has been showed was there are like C-Jun transcription factor. It interacts with the transcription factor. It's an accessory protein for the controlling transcriptional regulation.
Cofactor.
Yeah, cofactor.
It's possible that that doesn't directly bind to DNA.
No.
Yet you can pick it in ChIP-seq.
Correct.
Through some other protein.
Yes, also it's
so then you could do mass spec, right? Like co-IP mass spec to see what.
We can do those things, yes.
Okay.
So what is known already was RPL10 get activated by NIK kinase. The kinase is sitting in the outer cellular membrane. That transfers inside and that get phosphorylate. RPL10 get phosphorylate. That phosphorylated form goes into the nucleus, interacts with the transcription factor, activates the transcription of defense response genes. So very well studied functions have been known. In case of prostate, it's been very well studied with the CDCs, cyclin-dependent kinases for the cell proliferation. Yeah. Ribosomes.
Yeah, or chloroplast ribosomes.
That's a good question. The RPL10A1 and A2 are localized to the mitochondria and the chloroplast. Even though they are nuclear proteins, they are going into the mitochondria and chloroplast. And what was more intriguing was those proteins, although they are there, they're also localized to the nucleus. So there is what I'm assumption is they're involved in ribosome biogenesis mechanisms.
But both like cytosol and
yeah. Yeah. Thank you very much.
All right. Let's thank Ramu again. All right. So